Hi everyone, it's a pleasure for me to be here in virtually with you in this brilliant day that's been organised for you, um, ambitiously titled "Women of the Future," which I love, and I'm really sorry that I can't be there in person with you today. I've been asked to give a short talk on my research and also a little bit about my career. Um, and so it's going to be like an unusual one for me, and it's going to be a mixture of some of my research, which I'm not used to talking about in seminars, but then a little bit more about me, which I'm less used to going on about in seminars. So we'll see how it goes. My name's Ashleigh Griffin. I'm a evolutionary biologist. I'm actually my official title is Professor of Evolutionary Biology at University of Oxford, where I've been for about 10 years now. So I'm just gonna sort of take you through my sort of ideas and my research, what's inspired me, and how I got to the point where I am now. I've called the talk Meerkats to Microbes because one of the sort of fun things about being an evolutionary biologist is that there is you're studying a process, right? So you're not investigating a particular species of pathogen or an animal or a gene. It's a sort of underlying process that's common to all living things. And one of the fun bits of that is that you can be quite flexible about what you study in detail. So hence the sort of the meerkats to microbes theme, which is running throughout my research career. So this is a meerkat, and you're probably familiar with these from watching television documentaries. And one of the sort of things that they're really famous for, and what's made them really popular with public, is that they live in these like amazing family groups that you can see in this photograph. So they're a type of mongoose, and they live in the South Africa in the Kalahari Desert. And they're diurnal, so they move around during the day, where they're extremely vulnerable to their main predators, birds of prey. But they work together, so like they have individuals on guard duty, sort of like on the lookout for birds while they're all foraging. They forage with their heads underground, but burrowing for grubs, so they're really quite vulnerable while they're foraging. So they need sort of a guard to make sure that nothing dangerous is coming. So they work together, take turns on guard duty, and they also take turns on babysitting. So they have little pups that in the burrow, and then these pups are kind of naughty and want to wander off on their own, and they need to be looked after during the day while the group's out foraging, and they take turns in doing that. And the interesting thing about that is that the babysitter, so the individual that stays at home with the pups and doesn't eat anything in that day and loses weight, so there's a big cost to looking after the young, isn't the parents. So it tends to be like an older sibling, but it doesn't necessarily have to be an older sibling. Sometimes it's a completely unrelated individual. And so they're a really interesting species to study if you're interested in the evolution of cooperation. So how is it that some individuals pay a cost to perform a helpful behaviour for other individuals, when the benefit is not necessarily coming back to them? So what's in it for these babysitters? So there've been a big sort of study, the subject of a long-term study for the last 20-30 years or so, trying to understand how these cooperative behaviours evolve. And that's the project I worked on many years ago now, when I was a PhD student, and then at the University of Edinburgh. This is me in 1997, and as you can see there, meerkats are quite tame, so they're used to having researchers with them in the morning when they wake up at the burrow and following them around all day. And the field researchers have trained the meerkats to pop up onto these scales for a little bit of egg, and so that they can weigh them before and after the day's activities, and so get like incredibly like detailed information about an animal, a wild animal living in its natural habitat. So the specific task that I had when I was a PhD student is genotyping individuals in the group. So you can take like a little bit of skin and then like extract DNA from it, and use that DNA to tell whether they're related to one another. So you want to know if the helpers were related to the offspring that they're taking care of. So who are the parents of the pups? So that was my PhD work. And one, like I was saying, like the problem that the meerkats encapsulate, this sort of problem of cooperation. Why cooperate when you could selfishly use those resources to have your own offspring, is sort of central to understanding cooperation across the natural world, including humans. So in every case, whether it's these like cells here forming, these are like slime mold cells, amoebas that live independently, that come together to form these beautiful fruiting bodies. Now they cooperate to form these fruiting bodies, where cells up here will go on to form spores in the next generation, and cells that make up the stalk here are like a sort of an evolutionary dead end. So lifting up cells into the spore head, but these ones will die or be completely sterile. So that problem of cooperation is like encapsulated in this tiny little system here. We're more used to thinking about cooperation, of course, in terms of social insect nests, like these army ants. And it was like in humans, so like when we're trying to understand cooperative behaviour in humans, we're asking the same question. Why pay the cost if the benefit's going to other individuals? So by understanding cooperation in meerkats, we're sort of trying to provide we're interested in meerkats, but we're also trying to provide a sort of more general insights into how cooperation evolves in the natural world. So after my PhD, actually, like before I'd even finished my PhD, I got pregnant. So I thought it would be like I'm giving you like a sort of a snapshots of my life as I go through different stages of my research career. So just after I'd like finished with the meerkat PhD, I had my baby daughter Rowan, and I stayed. And after I'd graduated from my PhD, I didn't do any science for 3 years. I stayed at home to look after her full time. But when I was looking after her, I was sort of going over what I'd done in my PhD and thinking a lot about it. And over that time, I couldn't switch it off. I didn't sort of like stop thinking about science. And 2 research questions really sort of came through over that time. And I'm sorry it was cut off the text here for some reason. But the first question is like, so I found in meerkats that you had these unrelated helpers. So relatedness didn't really seem to matter too much. You didn't see the case where if a babysitter was more related to offspring, it would care more for those offspring or be more likely to go on babysitting duty. We didn't see that. But for other species, you definitely do see that. So other species, they only care if they're related to their young. And why is that? So one avenue of research that I wanted to go into is understanding the diversity of different cooperative behaviours that we see across animals and birds mainly. And for that, the approach that I took wanted to take, at that time I hadn't done it yet, was to use what we call a comparative approach, which is a picture on the right here, which is where you think about how traits have evolved over a family tree, and you can ask questions about the pattern of evolution over time. The second research question that sort of emerged from my PhD was thinking about, well, these meerkats help one another, but they're also competing with one another. And so what if that competition with one another negates the benefits they might have from helping one another. So in a way of putting that is saying, if you were to help your one sister have an extra offspring, if there's very intense competition, and there's only enough resources for one extra offspring, that might mean that another sister doesn't have an offspring. So when you have very, very strong competition, the relatedness doesn't really matter. You've got because it cancels out. So you're helping one relative, but that means that you're equally harming another. So is it the case that for meerkats, the competition was such that the effect of relatedness was negated? So I wanted to test the effect of competition on relatedness. You can't, the way to do that would be to set up two populations, one that's related and one that isn't, and see how much they cooperate with one another. And of course, you can't do that with meerkats. They're wild animals, and it would be unethical for a start, right? So starting to think about other systems, I'll come to this in a minute, this is where the microbes come in, but start to think about other systems that I could use to look at this question. So after my daughter was about 3, I started a postdoc. I managed to get some funding as a named postdoc, which means that I sort of come up with the ideas that went into the grant that first funded me. It was funded by the research council called the Natural Environment Research Council, and started to sort of had some funding to get started on testing my ideas, which is really, really exciting and thrilling. But there's a lot going on in my life at that time, so I was still very sort of, because I'd been with my daughter full time for 3 years, I found going back to work quite hard, and so I went back 2 days a week first of all, and then eventually 3 days a week, eventually 4 days a week, back to 3 days a week. So I was working part-time on and off for a long time. And I had another baby in the middle of my first postdoc. So this is another 12 months I had off to look after him. And so I was sort of trying to get things started, but at the same time, there's lots going on, and life getting in the way. So I just wanted to sort of tell you about these things because it's really important to understand. So before I go any further, I want to just emphasize to you that you might be thinking, why, and my children certainly ask me, why would you stop working on meerkats to work on bacteria? How boring is a bacteria compared to meerkat? Well, I can in some ways I can completely sympathize with that, but in other ways I would emphasize that you'd be surprised at how fascinating social behaviors in bacteria are. And in a sort of a slightly pathetic attempt to persuade you, I've got this video of some bacterial cells. So that white dot in the middle are some bacterial cells. Now watch this. So what you can see is a colony of bacteria growing with a fluorescent reporter gene in it, which glows fluorescent when a gene is expressed. Now that gene, if we go back, I can't go back. If that gene is a gene for a signal, right? So it's a gene that tells other individuals, cells around it, that it's there, and tells them to sort of switch on a whole bunch of different substances that might help them. So you can see that it's you can see that it's sort of, as soon as it hits the edge of the petri dish there, it starts the cascade of information being back. We've reached the edge. So there's it looks like a single organism, but it's not. It's millions and millions and millions of cells, individual cells, independent living, but communicating, cooperating. They're clearly coordinating their actions. So when back in 20 years ago, when I first started working on this, people thought that I was completely mad when I suggested that the ways that we think about meerkats might apply to bacteria. But at the same time, we're sort of wants to go ahead with this because, like I was saying, I want to do an experiment. And you can do some really cool experiments with bacteria. You do whatever you want with them. The way you don't have to worry about ethical considerations when you're putting together bacterial cells on a plate. So this sort of signal molecule is called Qscorum sensing, and the left-hand plate is a wild type. So that's a picture of the one that the same strain that I just showed you growing in time lapse, and the one on the right is a mutant that has that gene knocked out. Look at the difference between those two. The only difference between those two strains of bacteria is one can talk and the other one can't. Yeah. So when it can't, the colleague of mine that took this photo, he says the QS mutants just sit in the middle of the plate looking stupid. So just this dramatic difference that you can see when bacteria cooperate. So this is a wee cartoon to sort of explain that a little bit better. So on the left-hand panel here, we've got some bacterial cells coloured in blobs here. And they're making something. These, like, you can call them signalling molecules, or it might be an enzyme to break down proteins or proteins and make nutrients available. It makes these molecules and it excretes them to the environment. So bacteria have to digest all their nutrients outside of themselves. So we do that inside our stomachs, but bacterial cells' stomach linings are kind of like on the outside. You know, they have to sort of dissolve everything around about them. And to do that, they have to release enzymes into the environment. That's what these black dots are. Now you can see that, like, some of these cells are white, and they are the mutants, right, that don't make this stuff. So they're like the QS negative, the ones that aren't making it. They're in a mixture here with the producers. On the right-hand side panel, you can see that the white cells also benefit from the release of these enzymes, or whatever they are, signals. So you get a situation where you have some cells that are paying the cost of producing something that's helpful, but all of the cells benefiting from that helpful substance. And because they're not paying the cost, they have a fitness advantage, right? Because they're getting all the benefit without paying the cost. And so you can see an invasion of so-called cheats, individuals that are exploiting their neighbours, using up their enzymes which they have produced, which we sometimes call public goods. So they're like things that everybody can use, exploiting those and not contributing themselves. So it's not like a conscious thing. I'm using language that sort of implies that they're sort of consciously doing this. Of course they're not. They're just bacterial cells, but they just have a mutation that means that they don't make something, and that leads to them having more resources to divide and divide and divide, instead of having to put resources into producing the molecules, and so they spread. And you can use this system then to do experiments with cooperators who are making something and cheats that aren't. And so that's the system that I set up during my postdoc. So here is two cultures. The one on the left is a culture of a bacteria called Pseudomonas aeruginosa, which is a very nasty bacteria that is very dangerous if you've got certain conditions that depress your immune system. It's especially dangerous for people with cystic fibrosis. And it makes this green stuff called pyverdin, which is an iron chelator. So the black dot on the previous slide, it would be like something that binds iron, which is a really important element for metabolism of all living things. And it needs those things because when it's infecting a mammalian host, when it's inside a host, your hemoglobin and other molecules that you make to hold on to that iron, right? So if you get any infection, the bacteria has to sort of like steal your iron from a very, very sort of safe, tight bond with your hemoglobin. So that's what it's for. And some bacteria make it and some don't. The one on the right hand side, the white one, doesn't make it, so it's not great. And you can mix them together and then let them compete with one another, and then come back the next day and count the green ones and count the white ones. So you plate them out on agar like this, and you get these beautiful green colonies here, which are the colonies founded by a cell that's producing the pyverdin, and these little white dots, which are founded by cells that don't produce the pyverdin. You can count them. And you can calculate, like, so who's winning, who's got the higher fitness, so who's relatively good at dividing and producing copies of themselves. And your hypothesis is that if they're they're monoculture, so growing on their own, because they don't have that piiverdin, they're going to be less. The mutant is less fit than the wild type. So this one here is the equal fitness to the wild type. But if you mix them together, the mutant is more fit because it's able to do that exploitation and spread that I showed you earlier. So that's what you hypothesize. And that's exactly what we found. So when you have like high relatives, so that's essentially monoculture, the cooperators do better. To start off with like lots of little populations of 50% of which are only the piiverdin producers and 50% of which are only the mutants. And over time, you can see that the high relatedness, that cooperation goes to fixation. So all, by the time you've done this 4 or 5 cycles of competition, only cooperators are left. But if you have a mixture, so you allow them to compete with one another, you see that the cooperation is steadily declines, and there's a massive difference. So that relatedness difference is really key. And I haven't included the details now because I don't want to go into too many details in the short talk. So, but we also were able to answer the question that I talked about earlier about the scale of competition. So, I showed that in fact, yes. So, like if you do have really local competition, so really tight competition between cells, the effect of that relatedness disappears. So anyway, you can do like any number of experiments like this, which I did. What's the how does the relative numbers of copies and cheats affect this dynamic? How does the density of cells affect this dynamic? All of these things, and so I did lots of papers like that. But now, so just back to my career. So I imagine what it was like. So I had my baby. I had my first postdoc. I had another baby. I was working part-time. I was very tired a lot of the time, but I was really excited about my science, and I'd had lots of time to think about it, like I was saying. And so I did these 2 papers. One was looking at how important relatedness was in meerkats and other cooperative breeders, and the other one was looking at how relatedness was important in bacteria. And those 2, and I published one in Science and the other one in Nature. It's my first 2 papers that came out after I'd had my babies, which was at the time was amazing. And I've never been able to do that quite like that since. But what it meant is that I was able to continue getting money to continue science. So off the back of those papers, I managed to get like an amazing opportunity from the Royal Society, which is 5 years money, which is designed to help people who work part-time. It's called a Dorothy Hodgkin Research Fellowship. So it's perfect for me, and the start of a very long and supportive relationship with the Royal Society. So it's 2005 now. Like I said, I was doing lots of experiments, on bacteria, looking at the conditions that favoured cooperation. To be honest, it got a bit boring, and that's, you've got to sort of like listen to your boredom alarm when you're a scientist. If you get bored of something, it can be actually a good sign that you're, and you should listen to it. And so I got bored of it, and I think one of the reasons I got bored of it is because you sort of knew what was going to happen because you did the maths and you knew what the theory predicted. And so, surprise, surprise, when you went and put those things in flasks in a certain way, you got the results that the mathematics predicted. And I really was interested. What really motivated me was like finding out whether these social interactions affected bacteria in the real world, right? So infections. I started to get interested not just as in bacteria as a means to an end of testing theory, but as organisms in their own right. Okay, so where does Pseudomonas aeruginosa live? Well, they live in the human lung. It causes very serious disease there, and it's an environment about which we know the most of what their characteristics, just simply because it's highly studied, because it's an important pathogen in that context. So well, the first thing we did was just look at a number of these different products, like substances that the cells produce, to see what happened over time. And we looked at things like pyveridin, so like things that we're using in experiment, what happens to that in the lung. We looked at the signaling, which I showed you earlier, the Qscorum sensing signaling, what happens to that over time. And the enzyme for digesting proteins, what happens to that over time. And consistently across traits, we see this effect of decline over time. One of the consequences of not making proteases and quorum sensing signaling molecules and other things is that you're less harmful to the host, right? Because all of those things are like adaptations to get resources from your host, and we call them virulence factors. And you sort of like predict that cooperative bacteria then be more virulent than cheap bacteria. And so we did some experiments with a biologist called Kendra Rumble, who's based in Texas, where we looked at that in mice. And in this figure, you can see that these dotted line here is a mixed infection. These squares here are a quorum sensing mutant, and this is the wild type. So that you can see the wild type is much more deadly than the mutant, and that's simply because it's not making all of these other things that are harmful to the host. And so the next stage of my career is really sort of like trying to think more about like not just understanding bacteria social behaviour, but trying to harness it to think of next generation antimicrobial treatment strategies with antibiotics coming to the end of their shelf life in many cases. Can we use our understanding of evolutionary biology to provide another strategy? So coinciding with that kind of shift in focus in my research, I moved to Oxford about 10 years ago. I got continued funding from the Royal Society and a WotF, which is a fantastic freedom to just do research without any teaching or administrative responsibilities for 8 years. And then in 2014, I got my current position in the Department of Zoology. And I just sort of, so I became an established academic and with my own research group, and all of the sort of wonderful additional riches that brings. So here's me with my group getting news about getting a big grant. I now I'm a teacher, so I do a lot of undergraduate lectures, and I'm a tutor at New College, which I just absolutely love, and so I really enjoy that side of being an academic as well as the research. Not everyone does, but I do, and all the other sort of really fun things things you might not necessarily think of when you think of an academic career, going out talking to the public. Here's us in the Science Museum talking. We've made up a silly game to do with hula hoops and green tennis balls to explain our research to members of the public there. So now with the funding that I've got at the moment in the lab, one of the things that I'm doing, like I was saying, is trying to harness that social interactions to treat bacterial infection. So one of the ideas we have is it's called a Trojan horse idea, where we can use cheats. So the cheats are less virulent, but they're also good at invading, remember? So you can like turn a population of wild type into a population of cheats just by adding the cheats, if they can invade. And all those experiments, remember, that I did to sort of like work out when they're predicted to invade and when they're not. And so we can use all that information to make predictions about when invasion will be successful. And if we can do that, why not engineer those cheats to carry something like antibiotic sensitivity, or a suicide vector that sort of destroys cells in the neighbouring vicinity. So we can use the bacterial cells as like a Trojan horse that has like these medically beneficial alleles in it that we can use the cheating to force them through to population. So that's what I'm doing now. And you know, when like a lot of you be thinking, oh, I want to go into science to benefit mankind, and you know, and do something useful for society, and that's great. I would only say that like a lot of science and a lot of the beneficial, all the drugs and therapies and strategies that we have for combating climate change, all of those things, they all came from pure science originally, right? So you can't fix anything until you understand it. And so I hope that this talk has given you a sense of where like these sort of potentially applied ideas come from understanding basic processes in nature. And so we can draw this like direct line from developing novel treatment strategies against bacterial infection from meerkats. Great, okay, so that's a quick run through my life, my research, and I'm really sorry that I can't be there to meet you today, but I am going to be present for Q&A, I think, and of course you can just email me with any questions. That's azoo.ox.ac.uk cut off there at the end. Great, thanks.